Good evening students. In this video we are going to explore wonderful world of colors and we'll try to understand how exactly we perceive colors, right? For understanding color perception there are mainly two theories, are trichromatic theory and opponent theory, right? And these two theories don't antagonize each other, they kind of complement each other, right? They work at different levels of neural processing, right? So we'll just see, right, how these theories work and how overall our color perception functions, right? For understanding color perception, first of all we need to understand the trichromatic theory, right? For trichromatic theory, first of all we will draw a graph, right? Right, on x-axis we will have wavelength, right, 400 nanometer, (450 nanometer), 500 nanometer, 550 nanometer, and 600 nanometer. Right? Now, according to trichromatic theory, we have three types of cones, right? Red, blue, and green. In modern terminology, we call them L cone, M cone, and S cone, right? So at 560 nanometer, I'll draw a dotted line here, right? We are having spectrum of this absorption, right? So the L cone, it doesn't absorbs only 560 nanometer. It absorbs from a range of spectrum. All these cones have a range, right? Then we have got green cone, right, at 530 nanometer, then we have got a blue cone, 420 nanometer, right? So our L cone, also called red cone, right? This at 560 nanometer, our M cone, also called green cone, right, at 530 nanometer, our S cone, also called blue cone, at 420 nanometer, right? So these are three basic cones which we have got, right? Now we often call color as the brain's best guess, right? Because outer world is quite complicated, right? There are a whole spectrum of colors, but we have got only three cones, right? And with the help of these three cones, brain tries to guess the color, right? Now let's see. We have got L cone, M cone, and S cone. Now these two types of cones, your L cone and M cone, are the most numerous cones, right? If I go into the numbers, your L cone itself is like 65%, right? This itself is 65%, right? And M cone is around 30%, which leaves just 5% for your S cone. So S cone are least numerous cones, right? And you can see there is considerable overlap between these various types of cone, right? Now suppose if green light is coming, right? If green light is coming, it is not going to stimulate only one type of cone. It is going to stimulate some portion of blue cone also. It is going to stimulate some portion of green cone also. It is going to stimulate some portion of your red cone or L cone also, right? So suppose green light is there, right? It's going to maximally stimulate your green cone, like 60% of the green cones, right? But it's also going to stimulate some portion of your blue cone and some portion of your red cone, right? Since the green is getting maximally stimulated, so brain will perceive it as green, right? Be it any kind of cone, right? It knows that it is absorbing some kind of photon, right? It doesn't know what is the color of that photon, what is the wavelength of that photon, right? Only in the later processing inside the brain, right, the brain perceives, yes, these kind of cones have been maximally stimulated, right? So the color is this, right? Green light will stimulate all the three kind of cones, right? But the brain is going to compare, right, that which kind of cones have become maximally stimulated, right? And will try to guess the color according to that, right? That's why color is also called brain's best guess, right? Now, what these signals, let's suppose we draw some hypothetical cones over here, our blue cone, our green cone, our red cone, right? Now, where all this message from these cones will go, right? All this messaging from all these cones will go to the bipolar cells, right? It will go bipolar cells. Now they will converge on few bipolar cells, or they will, each cone will have its single bipolar cell, will depend on where it is located. In fovea, each cone will have its own bipolar cell because we have got high density of cones in fovea, right? Fovea has some peculiarity, we will come to it later, right? Now, these bipolar cells will further convey message to ganglion cell. Now, in ganglion cells, something interesting happens, right? Up to this point was our trichromatic theory, right? In ganglion cell, the opponent theory comes into effect, right? The ganglion cell theory was given by a scientist called Herring, right? He saw that reddish green color is not there, and even bluish yellow color is not there. So these two colors kind of oppose each other, right? So opponent processing happens at the level of ganglion cell, right? Opponent processing. Right? Now, our trichromatic theory, right, these three cones gives us the primary colors, red, green and blue, right? So, this trichromatic theory gives us primary colors. And what are our primary colors? Red, green and blue. Right? Now what do we mean by primary colors? That if we mix these three colors, red, green and blue, in equal proportions, we will get white color, right? And if we any kind of color can be produced by mixing these colors in different proportions. If we mix these colors in equal proportions, we will get white color. If we can get any kind of color we want by mixing these three colors in different proportions, right? So it's a very interesting concept of primary color, right? Your phone screen, right? It has got only red, green and blue pixels, but still your phone screen can produce millions of colors. An average human being can see something like 10 million colors, right? In humans, especially among females, we have got some tetrachromats, right? They can see all the way up to 100 million colors, but an average human being can see up to 10 million colors. So 10 million hues of colors are possible by mixing red, green and blue, right? And all your LCD screen, your mobile screens have got only red, blue and green pixels, right? And by mixing those red, blue and green pixels, we produce millions of colors, right? And what is the practical significance of ganglion cell open-end processing, right? The open-end processing in ganglion cells gives us our complementary colors, right? Complementary colors come from open-end processing in the ganglion cells. Complementary colors. Now what are our complementary colors? Our complementary colors are red and green. Right? Our complementary colors are blue and yellow. There are a lot of practical significance of these complementary colors, right? Suppose you want to highlight something, right? The background should be of the complementary colors. Suppose letters are red, the background should be of green color. Suppose letters are blue, the background should be of yellow color. Suppose the letters are yellow, the background should be of blue color, right? So complementary colors, right? When you want to highlight something, the background should be of complementary color. And when you mix two complementary colors, you get white color, right? But this complementarity is at the level of neural processing, right? But if you really want to mix the paints and get your white color, then it will slightly vary, right? Like if you want to get white color by mixing green with something, then instead of red you need purple, right? For mixing colors on a paint palette, it is going to slightly vary, right? Purple and green will give you white, right? And for getting white by mixing something with blue, right, you need orange. Blue plus orange, instead of yellow, slightly reddish, blue plus orange will give you white color, right? So it varies slightly when you actually mix the paints on a palette, right? So complementary colors have a lot of practical significance when you want to highlight something. And there is an interesting experiment also of complementary color, right? Suppose you focus on something red for a long time, like 30 seconds, right? Not very long time, just 30 seconds. And then you focus on a white screen. After focusing on something red for just 30 seconds, you focus on a white screen. You will get greenish hue. Why? Because the reddish cones have become fatigued in this time, right? And when you see something white, that red color is suppressed, and these red and green oppose each other. So the green color dominates when you see a completely white board, right? So red gets suppressed because of the fatigue due to continuous red stimulation, and when you see something white, right, you get greenish hue. The same stands true for blue and yellow, right? So complementarity arises because of the opponent processing in the ganglion cell, right? From the ganglion cell, right, it will go to the parvocellular layer of LGB, right? Parvocellular layer of LGB. From there, it will go to the visual cortex, right? Now, something interesting happens in visual cortex. As we discussed earlier, the brain actually assesses color with the help of comparison. If you stimulate only one cone, you won't see any color, right? At least two cones, stimulation of at least two cones is required for assessing colors, right? So in brain, we have got three pathways, right? We have got red-green pathway, right? Red-green pathway. What this red-green pathway will do, it is going to subtract the input of the L cone from the M cone. Red-green pathway, L minus M, right? Apart from this, you will have blue-yellow pathway. Right? What blue-yellow pathway will do, it is going to add the input of L and M cone and subtract it from the S cone, right? So in blue-yellow pathway, L plus M, right? Okay, minus S, right? There is another pathway which we call your luminescent pathway, right? Luminescent pathway. Luminescent pathway simply adds, it deals with the intensity of light. Luminescent pathway is going to simply add the L plus M cone. Ultimately, all this input will go to the V8 region of the cortex. Where further neural processing will happen. So knowing these three pathways is important because it tells us that brain actually compares the inputs of the various cones, right? If you stimulate only one cone, no color perception is there, right? At least two cones need to be stimulated, right? So brain has got three pathways, right? Red-green pathway, where it is going to subtract the input from the L cone, from the M cone. Blue-yellow pathway, it's going to add the input of the L cone and the M cone and subtract it from the blue cone, right? And the luminescent pathway, where it's going to simply add the stimulus of the L cone and the M cone, right? Now, how it will help in understanding color vision and how it will help in understanding color blindness. First of all, one very interesting example, right? Let's take the case of humans and our evolutionary close neighbors, be it humans, be it apes, be it monkeys, be it chimpanzees. All of us are trichromates. That means we have got three cones, L, M and S cone, right? And we needed this in evolution. But all the lower mammals, be it dogs, be it cats, be it deer, right? They are all dichromates, right? And there is evolutionary reason for this, right? The dogs, cats and deers and lions, right? Most of these are nocturnal animals, right? They hunt during night, right? So they've got only two cone systems, right? They are not color blind, but their visual world is not as rich as our visual world, right? When our ancestors were climbing on trees, right, they need to assess whether that fruit has ripened or not, right? For assessing whether that fruit is ripened or not, which cone we need, right? Fruits normally after ripening either become orange or yellow or red, right? On the higher wavelength side, right? Let me write Vibgyor over here, right? V I B G, okay, Y O R, right? So fruits after ripening either become yellow, orange or red, right? And this was of survival value, that an ape or a monkey should know when the fruit has ripened, right? So since this getting the cone which absorbs the wavelength on the higher side had a survival value, so we evolved that way, right? But the lower mammals, right, they need, for them the night vision is very important, right? So their retina is designed for night vision. They have a lot more rods compared to cones, right? So for them, the nocturnal, according to their nocturnal lifestyles, lot more rods are packed inside retina and cones are few. So due to the pressure of evolution, right, we got the higher, the cones having longer wavelength. So we are trichromates and lower mammals are dichromates. Now what difficulty it can produce for a deer? Suppose a deer is roaming inside a green grass, right, and a tiger, bright orange in color, right, or a lion walks through that green grass, right? Do you think that deer will be able to see that tiger or lion which is having yellow or orange color, right, through the green grass? A human will be able to see. A human can easily assess that yes, in that green grass we have got a yellowish tiger. But for a deer, right, he can see only up to this point, right? So even that tiger will appear greenish to that deer. So it's an interesting example of how our visual world is different from the deer's visual world, right? So if we take the examples of trichromates, who are all trichromates, we know, right? Humans, apes and monkeys. And monochromates, which dichromates, which dichromates we know? Cats, deer, cats, dog, deer, horses. Right? So it's an interesting example of. One more thing I wanted to discuss was the case of Foveal, right? This Foveal has got only L cones and M cones, right? This fovea has got only L cones and M cones and no blue cones, right? Just think of it. Your center of perception, the point where you get the finest details, have got no blue cones, right? And this creates many practical issues, right? If something is written in very small letters, right? Suppose something is written in very small letters in red and something is written in very small letters in blue, right? You are going to read relatively easily that thing written in red letters compared to blue letters, right? And this is the reason fovea has got no blue cones, right? So if something is written in very small letters in blue, it's tough to read it because center of your vision has got no blue cones. But we can conduct a thought experiment. Suppose there is one big blue screen, right? If there is a one big blue screen, why don't we see a whitish dot at the center of that blue screen, right? If we have got no blue cones in the center. This is because the brain continuously fills the information inside that spot, taking the information from the surrounding, right? This is very similar to the blind spot. All of us carry a blind spot 12 to 15 degrees temporal to our center of vision, but we never see it because brain is continuously filling that blind spot, right? Similarly, we have got a blind spot for blue color at the center of our vision, right? But we don't see that blind spot for blue color because brain is continuously filling the information by using the information from the surrounding, right? So this is a very interesting example of how exactly, right, our center of vision lacks the blue cones, right? So fovea has got only L cones and M cones. No blue cones. But then we should ask why nature has designed the whole thing this way, that we have got no blue cones at the center of vision, right? This is because to avoid chromatic aberration, right? Your blue light focuses in a slightly different plane compared to your red light and green light, right? Blue light focuses in slightly different plane. By eliminating blue light, we bring lot more clarity to the vision. Lot more clarity comes in vision when you eliminate blue light. Lot more chromatic aberration is avoided, right? So nature has done it to bring lot more clarity so that you can see lot more finer details in fovea, right? But the side effect is that if something blue is written in very small letters, it's tough to read those letters, right? Apart from that, we can discuss color blindness, right? In color blindness, right, we have got trichromates, we have got dichromates, and we have got monochromates, right? If in trichromates, all the three cone systems are present, but one cone system is weak, right? So if something happens, if one cone system is weak, we will always call it anomaly, prota anomaly, deutera anomaly, trita anomaly, right? If something happens to red cone, we will call it prota anomaly. If something happens to green cone, we will call it deutera anomaly. If something happens to blue cone, we will call it trita anomaly, right? Apart from that, we have got what we call dichromates. In dichromates, both the cone systems, right, two cone systems are present and one cone system is absent. Since one cone system is absent, we will call it anopia, protanopia, deuteranopia, tritanopia, right? The deutera anomaly and deuteranopia, right, are two most common. And in monochromates, what will happen? They will see one color? No, they won't see any color, because when only one cone is stimulated, right, in that case you won't see any color, right? Why? Because brain constructs color by comparing the input from the different cones. Cones doesn't know which wavelength it is absorbed, right? It is for the brain to interpret that which kind of cones have got maximally stimulated, and brain creates and construct color based on this, right? Long time ago I read a thought that before brain there was no color, no sound, right? There's nothing like color in the outer nature. Your brain construct color. There's nothing like sound in the outer nature. Your brain construct sound. Same way, right? So your brain construct color by comparing the input it receives from all the cones, right? So hopefully I was able to explain the color vision, right? I'll just sum it up, right? One very interesting thing which you can see over here is that when this red has got maximum absorption, you get yellow, right? So in some books, right, you will see that red cone is called as yellow cone, right? Red cone is often called yellow cone. And to avoid this confusion, we are now calling it L cone, M cone and S cone, right? Because they are not absorbing color of any single color, they have got a range, right? Just the spectrum of that range is different, right? This gives us also a very important, interesting concept of metamers, right? Now, what is the concept of metamers? Metamers is that suppose you focus, a pure yellow light of 580 nanometers on someone's eyes. He will see it as yellow, right? But then you give him a mixture of red and green light, right? Now, the pattern of cone excitation produced by that pure yellow light and the combination of the red and green light is same, right? So a spectrophotometer might detect that yes, this is pure yellow light and this is combination of red and green light, but for your own eyes, right, it will appear exactly the same. And this trick is used by mobile companies, LCD companies, right? They mix the red and green light to make it yellow. They don't have any yellow pixels on their screen, right? That's a very interesting example of metamers. How exactly through technology we fool our senses, right? The pure yellow light and combination of red and green light will give us the same input, right? Because brain is not detecting the color exactly, brain is just detecting what kind of cones have been stimulated. If yellow light and red and green light stimulate the same kind of cones, brain will interpret it as yellow light, right? So there are many interesting things which we discussed in color vision, right? So what are the two theories with the help of which we can explain color vision, right? Trichromatic theory and opponent theory. In trichromatic theory, we have got three kinds of cones, right? L cone, M cone and S cone, right? L cone has absorbs light of highest wavelength 560 nanometer and it is the most numerous. M cone absorbs light of 530 nanometer and it is just 30%. S cone is least numerous. It absorbs light, peak absorption at 420 nanometer. They will convey all this information to bipolar cells and they will convey all this information to ganglion cells where opponent processing happens, which gives us our complementary colors like red and green and blue and yellow. Our primary colors are red, green and blue. If we mix these colors in equal proportion, we get white. If we mix these colors in varying proportions, we can get any color out of the 10 million hues of colors which we can see, right? Humans, apes and monkeys are trichromates. Cats, dogs and deer, right, are dichromates. They find it tough to see the wavelength on the higher side. So it's slightly tough for them to see yellow, orange and red color. Fovea has got only L and M cones. They have got no blue cones. That's why we find it tough to read, right? Small letters written in blue color, right? As we said, in visual cortex, we have got three kind of pathways: red-green pathway, blue-yellow pathway and luminescent pathway. Red-green pathway will subtract, right, the input from the red cone from the green cone, L minus M, right? Blue-yellow pathway will add the input from the L and M cone and subtract it from the S cone. Luminescent pathway will add the input from the L and M cone, right? And we also discussed about the color blindness, right? Trichromates, dichromates and monochromates, right? In trichromates, all the three cone systems are there, but one is defective. So we will use anomaly in this case. Prota anomaly for red, deutera anomaly for green, treta anomaly for blue, right? In case of our dichromates, two cone systems are present, but one system is absent, right? So we will use anopia in this case. Protanopia for red, right? Our deuteranopia for green, tritanopia for blue. Monochromates, no color vision, right? So hopefully this video has helped you, and hopefully you have enjoyed this video, right? Hopefully this video has helped you in better understanding of color vision, and hopefully it has stirred the curiosity inside you to explore the world of colors, right? Wish you all the very best. Thank you.